For so many families, a rare genetic disease is this painful mystery with no clear answers. A child has a condition that doctors just can't put a name to, and it kicks off this long, often frustrating journey to get a diagnosis. But now, a new wave of genomic science is really starting to turn the tide. Today, we're going to look at how these amazing scientific detectives are finally cracking the code, and what they're fighting is, honestly, truly remarkable.
So you probably think of rare diseases as, well, rare, right? But here's the thing that might surprise you. When you group them all together, they're actually incredibly common. Worldwide, they affect about 1 in every 20 people. Just think about that. It means in your office, on your commute, or even in your own extended family, there's a really good chance someone is affected by a rare disease.
But here's the real challenge. For more than half of these people, the search for a genetic cause, it fails. The tests come back with nothing, leaving them and their families in this awful state of uncertainty. They're on what's called a diagnostic odyssey, and it can go on for years and years.
So, in this explainer, we're going to break down how scientists are finally starting to tackle this head-on. We'll start with the puzzle itself, then introduce the incredible team that's been assembled to solve it. We'll look at their brand new toolkit, see how they're cracking the absolute hardest cases, and then wrap up by exploring their vision for a shared genomic future.
Okay, so let's start with the problem. This whole diagnostic odyssey thing, it's not just frustrating for families, it's a massive scientific hurdle. So why is it so incredibly hard to find the genetic smoking gun for so many of these conditions?
Well, the answer is complicated. As you can see here, the challenges are coming from all sides. The genetic typo might be hiding in a gene we haven't even connected to a disease yet. Or it could be lurking somewhere in the other 98% of our DNA that doesn't code for proteins, the part we used to call junk DNA. And sometimes the very technology we're using just isn't up to the task, making certain types of genetic mistakes practically invisible. So when you're faced with a puzzle this huge, no single lab, no single researcher can solve it alone. And that's where the hero of our story comes in. A massive, coordinated effort designed specifically to take on these unsolved cases.
Meet the GREGoR Consortium. It was launched back in 2021 by the National Human Genome Research Institute, and its mission is both simple and incredibly ambitious. Bring together the best and brightest minds, use the absolute newest technologies, and, this is key, share everything they find to speed up discoveries for everyone.
Now, this slide is really cool because it basically lays out their battle plan. And you can see it's a full-on, multi-pronged attack. It starts with systematic data generation. That means using some seriously powerful new sequencing methods. Then you have computational innovation, so building new tools to actually make sense of all that data. And the whole thing is built on this foundation of data sharing and a commitment to what they call genomics for all. The end game, to dramatically boost the number of successful diagnoses and finally put an end to those diagnostic odysseys.
So what are these powerful new technologies they're using? Let's open up GREGoR's genomic toolkit and see how they're able to find things that were completely invisible before.
Okay, so for a long time, the standard method has been something called short-read sequencing. The best way to think about it is like trying to read a book by looking at just one word at a time. You'd be great at catching typos, but you might totally miss that an entire paragraph was copied and pasted in the wrong place. Well, long-read sequencing is like being able to read whole paragraphs or even pages at once. It lets scientists spot these huge, complex structural changes in the genome that short reads just completely miss. It's a total game changer.
And it's not just about the DNA anymore. The really crucial piece is figuring out what that DNA is actually doing. And that's where multi-omics comes in. It's all about getting this 360-degree view of a patient's biology by layering in data from RNA, which carries the instructions from DNA, and even from proteins. It helps connect the dots from a genetic variant all the way to its real-world effect on the body.
And the scale of this whole effort is just wow. It's massive. This table gives you just a little snapshot. We're talking about thousands of people getting their entire genome sequenced with short reads, hundreds with those powerful long reads, plus hundreds more providing RNA and other types of omic data. This is creating a scientific resource that is just completely unprecedented.
Here's another really clever part of their strategy. They're not just looking at the person with a condition. The majority of the time, we're talking 59%, they're studying what are called trios. That means the patient and both of their biological parents. By comparing the child's genome to their parents', scientists can spot brand new mutations, what they call de novo mutations, that popped up for the first time in the child. And often, that's exactly where the answer lies.
So, we've met the team, we've seen the toolkit, now for the exciting part. Let's look at how this all comes together to solve some of the most challenging mysteries in all of genomics.
This quote from the source material really gets to the core of what GREGoR is all about. They are not looking for the easy wins, the low-hanging fruit. No, they are specifically going after the cases that have stumped everyone else, the ones where all the standard tests came back empty.
So what do these hardest cases actually look like? Well, you can think of it like a series of detective missions. They're hunting for clues in that vast, non-coding genome. They're using long-read sequencing to entangle these massive, complex scrambles of DNA. They're decoding these really unstable, stuttering sections of DNA repeats. And they're finally getting evidence to make a call on variants of uncertain significance, or VUSs, which are basically the genetic equivalent of a question mark.
And this new approach is already paying off with some absolutely incredible breakthroughs. So get this. By digging into these unsolved cases, GRER and their collaborators discovered that mutations in a non-coding gene, a gene called RNU42, are actually one of the most common single-gene causes of certain neurodevelopmental disorders. This is a discovery that just wasn't possible without this new toolkit.
And this all leads us to the final and maybe the most important part of this whole story. The fundamental belief that this science has to be a shared project for all of humanity.
See, GREGoR operates on this principle of what you might call radical collaboration. Their data isn't locked away on some private server. Nope. All the genomic information from over 3,000 families is made available, and quickly, to any qualified researcher anywhere in the world. The whole idea is to basically crowdsource the hunt for answers and accelerate progress for everybody.
And this spirit of sharing goes even deeper, all the way to equity. You see, for way too long, genomics research has mostly focused on people of European ancestry. GREGoR is actively working to change that. They're recruiting diverse participants, they're building tools that work better for people of all ancestries, and they're tackling that problem of USVS, which are way more common in non-European populations. The goal, in the end, is truly genomics for all.
But even with all this incredible progress, the work is nowhere near finished. This slide gives us a peek at the next great frontiers. A massive goal is to finally complete the entire catalog of all human disease genes. But beyond that, scientists are just starting to figure out how combinations of variants in different genes might team up to cause disease, which opens up a whole new universe of complexity.
Which leaves us with one final, fascinating question to think about. For decades, the goal has been to find all the genes linked to human disease, and every day we're getting closer. So, what happens when we finally finish that catalog? What will medicine even look like when we hold the complete instruction manual for human health and disease?